Hello, everyone, and welcome to today's webinar, Gibco Cell Culture Hero webinar, Interrogating Cardiac Cell States with Genetically Encoded Reporters in Human Embryonic Stem Cells. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speaker for today's webinar, Mick Lee, PhD, Research Fellow from the Cardiovascular Research Institute, Department of Medicine. Mick, you may now begin your presentation.
So thank you for the kind introduction, Sydney. So first and foremost, I would like to thank the team at Thermo Fisher and the Gibco team for the kind invitation for us to share about our work on cardiomyocyte cell fate and cell states. I'm a research fellow, one of the senior fellows at Professor Roger Fu's laboratory at the Cardiovascular Metabolic Disease Translational Research Program at the National University of Singapore, and also within NUS Cardiovascular Research Institute. And the agenda of this webinar is to really discuss about what we have done so far and generally in our efforts to use a genetic-based approach to interrogate cardiac cell states, such as using CRISPR screening approaches and leveraging on reporter systems for gene discovery and validation. And this application can be quite varying as well and be applied to other fields. And in particular, we are interested in understanding gene regulation in cardiomyocytes and reprogramming them to regenerate for therapy. And we are now looking at cardiomyocyte as post-mitotic cells don't proliferate. This is a problem in end-stage heart failure. And I have always wanted to understand how genes are regulated and how chromatins are wired differently in different cell types. And even though every cell is encoded by the same genetic makeup, as that's how I got started with epigenetics at the Genome Institute of Singapore when I first started my PhD. And however, how the way the cell reads the DNA is different. The use of enhancers are regulated very differently depending on cell states and disease status. So I spent my PhD training in the Genome Institute of Singapore, and one of the projects I was working on was on gene regulation and epigenetics in the heart, as well as using CRISPR to validate the role of a list of carefully curated heart disease-associated enhancers and genes. And I was also asking if genes and chromatin wiring affect cell states as cardiomyocytes can become mature, disease, or reprogrammed. And then really the question to ask if there are really ways to functionalize the role of these genes and known coding genome using a high-dimensional CRISPR screen. At Professor Roger Fu Lab at the National University of Singapore, we are committed and we are interested in using multi-omics and induced pluripotent stem cells to study cardiovascular metabolic disease, and also to understand the impact of genetic variation in health and in disease. We are also a team of scientists working on different teams around this topic, and we are also recruiting talented postdocs and young aspiring faculty members to the Translational Research Program. So coming back to the treatment options for heart failure, in the past decades, by practice, there is two main class of foundational drugs, the beta blockers and ACE inhibitors, just to name a few, which were given to mitigate adverse cardiac remodeling. And these are basically drugs that stop the heart from working too hard and wearing out, especially for heart failure with reduced ejection fraction. However, these are not really solving the root cause of the problem, in which cardiomyocytes, when they are lost through cell death, cannot be replaced endogenously. And the current replacement therapies are LVADs, left ventricle assisted device, and transplants. However, limited by donor availability, and the LVADs are not sustainable because there's limited mechanical lifespan. Of course, there are now new ways, stem cell-based remuscularization strategies on the horizon, which we are also very excited about. And of course, harnessing the intrinsic capability of cardiomyocytes to divide again, which I think is the holy grail. So cardiomyocyte regeneration is limited to early postnatal life, as demonstrated a decade ago, and the molecular cues that induce cell cycle arrest in cardiomyocytes to become terminally differentiated cells still remain somewhat obscure. And the reason why cardiomyocyte lack this intrinsic ability to proliferate, there are several reasons. Number one, metabolic switch from glycolysis to fatty acid oxidation to meet higher energy demand creates a lot of reactive oxygen species and mitochondrial content, induce DNA damage, and that reinforces cell cycle arrest, epigenetic repression of some of these cell cycle genes, and loss of some of these key signaling developmental cues, just to name a few. And of course, we are now beginning to understand the main signaling pathways and regulators that may be controlling the ability of cardiomyocytes to proliferate. And the key is to activate or repress some of these drivers or roadblocks. And this encompasses microRNAs, the hippo-ERBB2 signaling pathway, for example, and the extracellular matrix components and factors as shown by many groups over the past decade. And tweaking these pathways may hold key to harnessing the endogenous capacity of the heart to repair itself. On the other hand, another strategy would be to use of cardiac patches and scaffolds carrying exogenous iPSC-derived cardiomyocytes to support this new vascularization and patching of the heart. And for us, we want to start by asking the question, what makes a cardiomyocyte? And here, using the epigenetic Weddington landscape, which a lot of us are familiar about, to illustrate here, as the pluripotent stem cell begins as undifferentiated cells and move down the slope of increasing cell-fate restrictive intermediates and forming terminally differentiated cells like cardiomyocyte, we then ask, what are the regulators of cardiac differentiation? And if we can also ask the question, what are the regulators of dedifferentiation and redifferentiation? So under specific signaling cues, we can differentiate iPS cells into their respective terminal lineages. And it turns out that cells at the valley can also be altered through reprogramming, dedifferentiation, or transdifferentiation, either from one cell type to another, or first converting cells into a less mature multipotent cells, which is a process of dedifferentiation, and then taking the step back to allow regeneration to take place before redifferentiating back to mature cells. So I would like to take this building analogy as an example. It's like tearing down an old building and then getting new workers to build more new buildings on the same piece of land. So like cardiomyocytes, they bear structural constraints as mature cardiomyocytes are packed with sarcomeres, which physically limits the nuclear reorganization and mitosis. So these sarcomeres needs to be broken down and revert back to a fetal-like state for cell cycle re-entry to happen. So to understand what makes a cardiomyocyte, here we use an MYH6, myosin heavy chain 6 GFP reporter line to sort for cardiomyocyte in the non-myocyte population and perform a genome-wide CRISPR screen using the human whole genome knockout Brunello library in the pool of Cas9 expressing stem cells, subjected under the classical cardiomyocyte GV differentiation protocol by Wnt signaling modulation. And by day 14, the cells were then fact sorted, and following which the differential guide RNA enriched in the non-myocyte population was identified. And we highlighted several known genes in the scatter plot on the bottom right-hand corner, as you can see, that some of these genes implicated in activin signaling pathway, cause-wise SNP components, and some of these known developmental pathways of genes are highlighted. And here we identified NF2 to be a known, to be an upstream regulator of the hippo-YAP pathway to take it to study further. And we then perform transcriptome sequencing across the differentiation time point of the wild type and NF2 deleted cells, which we observe a clear bifurcation of cellular trajectories as early as day 1 to 3 mesendoderm. And the knockout cells were then left to culture to continue to proliferate. And to discern the terminal cell identity of the knockout cells, we prioritize the key regulatory factors through the RNA-seq data by cell type specificity based off the encode-based epigenetic K27 ME3 repressive domains using the triage tool from Dr. Nathan Palpin's group at the UQ. for which we identified the presence of epithelial marks and anti-cardiac regulators prioritized in the knockout cells. This suggests that the NF2 knockout cells have adopted a radical shift in identity towards more of the epithelial-like lineage. And here you can see clearly the impairment of epithelial mesenchymal transition at day 2 in the knockout cells, and that further led by the ectopic expression of CDX2. And using the cardiac differentiation protocol and Western blot across time points in the NF2 deleted cells, we also observe the loss of eumesodermin and some of these early lineage-specific markers. More significantly, we observe that there is no change in phosphorylation status in the core hipokinases, that's marked in red, despite the loss of phosphorylated YAP just above these red boxes here. And you can see that the OCT4, one of the anti-cardiac repressors, remained upregulated from day 5 onwards. We suggest that NF2 may be regulating YAP through a non-canonical pathway. And here you can see that we observe the loss of phosphorylated angiomotin, specifically at day 1 mesoendoderm in the knockout cells. We suggest NF2 may be regulating YAP via angiomotin, independent of the canonical core hypo-YAP signaling as early as at the mesoendoderm stage. In the NF2 deleted cells, we can see that the YAP remains in the nucleus during early epithelial mesenchymal transition. That suggests that it is repressing these early mesoendoderm genes. And this is also marked by the lack of removal of repressive roadblocks like OCT4, SOX2, and CDX2, and MSX1 in the knockout cells. And in summary, and taking everything together, you can see that the knockout cells undergo a radical shift in identity towards epithelial-like cells, and altogether, the de-repression of YAP is necessary during early cardiac differentiation, and its localization may also be the answer to the cardiomyocyte ability to proliferate. And here we observe that the subcellular localization of YAP is very dynamic, especially during cardiomyocyte differentiation and development, as it exits the nucleus fairly early during differentiation. And in early neonatal cardiomyocytes to adult stage, you can see the transition of YAP localization predominantly outside of the nucleus. And hippo pathway inactivation has been shown in numerous literature to induce cardiomyocyte cell cycle re-entry by the work of James Martin's group at the Bayer College of Medicine, that's Zahor's group at the Weizmann Institute on the ERBB2, and others showing that it is protective against myocardial infarction. And this has been also shown that this phosphorylated YAP is also very much phosphorylated. So in the failing heart, you can see there's more cytoplasmic YAP than in the nuclear YAP, and suggests that hippo pathway inactivation can induce some form of cardiomyocyte regeneration. And to study the journey back up the hill, a postdoc in our lab, Ike, has utilized an orthogonal in vitro and in vivo murine model to model cardiomyocyte de-differentiation. And the in vitro model on the left shows how adult murine cardiomyocyte that were primarily rod-shaped loses its sarcomeric organization and integrity as soon as it is cultured on plasticware, and then it reforms the sarcomeres again by day 14. The cardiomyocytes are no longer rod-like in shape, but rather it adopted more of a neonatal-like morphology as if they have lost their maturity. So we performed RNA-seq across these different time points and identified these de-differentiation markers that peak between day 4 and day 7. That's the point where cardiomyocytes break down the sarcomeres and then they start rebuilding again. And separately, in collaboration with Thomas Brown's group and Johnny Kim and Yen Pu at the Max Planck Institute, we also used their doxycycline-inducible Yamanaka OSKM mice model for transient cardiac reprogramming to push cardiomyocytes into a more plastic regenerative state, to which Ike was able to see a modest cardiomyocyte proliferation and regeneration. So this mice model is great because it allows for a conditional, inducible, and exchangeable model for studying cardiomyocyte de-differentiation. As there isn't really a robust marker for cardiomyocyte dedifferentiation, ECA has overlapped both the transcriptomics data from the in vitro and in vivo model, which revealed ASNS as a sole candidate, which stands for asparagine synthetase. So ASNS converts aspartate and glutamine into asparagine, and asparagine has been shown previously to activate the mTOR pathway for anabolic, metabolic, and cell growth in cancer cells. And what she has observed is that these ASNS positive cardiomyocytes in both models are shown to have more disorganized appearance in sarcomeres than the ASNS negative cardiomyocytes. And these cardiomyocytes also marked by proliferative signals that is marked by Ki67. And now when she knocked down ASNS with the overexpression of the Yamanaka 4 factors, she see that there are lesser and fewer dividing cardiomyocytes. We also overexpress an activated version of YAP5SA using the adeno-associated virus. And here we see an expected increase in this PH3 proliferating marker, as well as the enhancement of ASNS expression level. And finally, we also looked at the downstream mTOR kinase and the binding proteins and observed these reduced downstream mTOR signaling marked by S6 kinase. Which we then think that ASNS is necessary for sustaining mTOR in dedifferentiating cardiomyocyte necessary for survival and cell cycle re-entry. So ASNS is an essential mediator of cardiomyocyte dedifferentiation, regulating asparagine metabolism, while playing distinct role in vitro and in vivo. And when knocked down, the mTOR signaling downstream was reduced, particularly the S6 kinase. And the asparagine synthase is necessary to sustain this mTOR activation, a potential regulator of cardiomyocyte survival and cell cycle during cardiomyocyte dedifferentiation. So we asked if it is possible to engineer an ASNS reporter system as a readout to screen for regulators of cardiomyocyte dedifferentiation. So one problem with iPS and human embryonic stem cells is the transgene shutdown when using lentivirus or piggyback system upon terminal differentiation. You can see that the shutting down of the Cas9 protein across cardiomyocyte differentiation was observed here following maturity by day 30. And using a transposase safe harbor knock-in approach, we can now efficiently knock in any transgene or payload with a landing pad and the BXB1 recombinase, targeting to a single locus through the use of any transfection method. And the work of this work is led by this, one of the senior scientists in the lab, Matthias, and we have now been able to leverage on genetically encoded tools to knock in these transgene and reporter into the safe harbor. And how we also managed to knock in the CRISPR system into the safe harbor for transgene and gene repression in terminally differentiated cells. And we were also pleased to improve our knock-in efficiency with our new Neon NXT electroporator with the help of the Thermo Fisher team as well. We managed to get an improvement of knock-in efficiency to approximately about 50% using a dominant negative P53 and the BXB1 recombinase and the payload, compared to the existing transfection method that we have been using in the lab using the lipostem fact method. And we have been able to achieve consistent knock-in within the single locus in less than a week using this approach. We have also tested different promoters, including the human ASNS promoter, and we see that the CAC and the ASNS promoter robustly switches on the GFP expression when knocked into the safe harbor, but this is not seen in the cardiac troponin promoter, which we use as a negative control. And interestingly, we see that the EF1 alpha promoter appears to show signs of promoter shutdown after 6 passages, suggesting that the promoter actually does matter when doing any experiments with human embryonic stem cells or human iPSCs. And now that we have identified the ASNS as a marker for cardiomyocyte dedifferentiation, we went on to create this ASNS GFP reporter line using human embryonic stem cells, which we then direct its differentiation into cardiomyocyte. And we test this response across the different stimuli and metabolic cocktails. And as expected, these ASNS level decrease upon differentiation into cardiomyocyte, and the signal decreases when they are placed in the maturation media. And using the real-time reporter system, and we are able to perform a genome-wide CRISPR screen using the same library earlier, the human genome-wide Brunello knockout library, which we then use to identify genetic regulators of ASNS. And as expected, mTOR pathways and its regulators like TSC1 and TSC2 were highly enriched in the top list of genes for the loss of function mutation on these genes that increase these ASNS levels. So we are really excited to pursue some of these candidates further, and we'll give the community further updates in time to come. So in summary, for the first part of this webinar, we have performed a genome-wide CRISPR screen using a cardiomyocyte-specific MYT6 reporter to identify regulators of cardiomyocyte differentiation. And the hippo effector YAP promotes cardiomyocyte proliferation, typically when it gets, the hippo pathway gets inactivated in the mature adult cardiomyocyte. And the ASNS, which we have identified as a marker for cardiomyocyte dedifferentiation, is regulating asparagine metabolism, and it also plays a distinct role in vitro and in vivo as well. And ASNS also plays an important role in sustaining the mTOR signaling pathway, a potential regulator of cardiomyocyte survival and cell cycle during cardiomyocyte dedifferentiation. And we also perform another genome-wide CRISPR screen to identify the top regulators of ASNS to establish. And then we also have established these reporter lines for further investigation. And hopefully this could be then be used for targeted for regenerative therapy. In the next 10 minutes or so, we'll discuss a little bit about 3D organoid system and the cellular crosstalk of cardiovascular cell type. Starting from the epicardium, which is a layer of mesothelius tissue that envelopes all vertebrate hearts, has emerged as a recent player in cardiac repair and regeneration. And the epicardium is essential for muscle regeneration in the zebrafish model of innate heart regeneration, and it also participates in fibrotic responses of mammalian hearts as well. And this structure serves as a source of key cells like vascular smooth muscle pericytes and fibroblasts during heart development and repair. And it also secretes some of these growth factors that are essential for proliferation and survival of cardiomyocyte. And activating this adult epicardium for heart repair also requires directing this tissue to promote neovascularization rather than scarring, and to function as a pro-regenerative rather than pro-inflammatory. So this epicardium-myocardium signaling plays a very important role in mammalian heart repair. Additionally, on the left side of this slide, you can see that macrophage also plays an important role as it surrounds dedifferentiating cardiomyocyte, and that macrophages also contribute to ECM remodeling at border zone post-injury, and it also promotes cell migration and invasion in regenerating cardiomyocyte to replenish these lost cells in ECM-laden tissue. And cardiomyocyte and fibroblast macrophage crosstalk and this cellular triad has been shown previously by the work of Jim Martin's group and Rich at the Baylor College of Medicine. And given the importance of stromal cells and immune cells, we are now able to establish some of these protocols in the lab in-house, differentiate iPS cells in human embryonic stem cells into major cardiovascular cell types using directed differentiation for study. So Matt and his team has very much diligently able to set these protocols and optimize them in the lab here at the Cardiovascular Research Institute. And in 2021, that's when cardiac organoid really come to the fore and into spotlight, with many self-assembly approaches starting from EB, with organoids forming chambers. But most existing cardiac organoid protocol lack immune cell fate because the macrophage population is derived from a different hemogenic mesodermal lineage. And using this direct assembly approach, it is possible to independently differentiate respective cell types and then assemble together with varying cell populations or proportion to form these cardiac organoids or cardiac microtissues. So following differentiation into terminal lineages, our 3D cardiac microtissue model encapsulates the 4 cell type, and that includes the macrophage now. And leveraging on the safe harbors and genetically encoded reporter systems such as the calcium reporter, the ATP sensors, as well as the Fuji system, we can obtain real-time reporter phenotypic readouts for downstream high-content imaging and fact sorting. And whether it involves gene knockout or overexpression, or the introduction of a reporter system, this can now be validated in a system comprising the 4 major cardiac cell types, thereby revealing some of these phenotypes that are typically not observable in the 2D setting. And this is also generally inspired by a compendium of genetically encoded biosensors for real-time qualitative imaging, which we are now knocking them into the safe harbor cells developed by Matthias. And here's an example of the GChem6F reporter line. The video is not working over here due to some technical difficulty, but rest assured that this GChem6F reporter line is flashing and in the 3D cardiac microtissue model. And we can see that the 4 cell type cardiac microtissue model is very much compact, and under the high fatty acid and maturation media cocktail, we're able to show this enhanced contraction and calcium handling that is indicative of a more mature cardiac microtissue. And also leveraging on the ATP reporter system, we also show how the ATP sensor is responsive to the ATP stimulus, and also under high fat and high sugar cocktail, and that also coupled with the impairment of the contractile function. And so we are really excited to scale up these 3D cardiac microtissue now, and leveraging a suite of genetically encoded reporter system for high content imaging to better model and study cell-to-cell crosstalk in the context of cardiac regeneration and disease. And in summary, for the second part, we have performed multi-cellular 3D cardiac microtissue and organoid models to engineer physiologically relevant cardiac models for disease modeling and studying cardiovascular cell state and niches. We have also engineered multimodal genetically encoded reporters into safe harbors, as well as that basically allows for more robust and safe transgene expression. And we are still in the process of knocking more reporters into this system, and hopefully it could be used widely in the community. And we are also knocking in these reporters for scalability and batch-to-batch reproducibility without the use of viral delivery systems. And with that, I would like to thank my lab members, especially Ika, Matthias, and Matt, who have been instrumental in driving various aspects of the work, as well as our fellow collaborators for their unwavering support in our endeavors. And I would like to once again thank the Thermo Fisher team, the LabRoots team, and the team for their support in organizing this webinar. And I am very happy to take any further questions. Thank you.
Thank you, Mick, for your very informative presentation. We will now move into the Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just type your question in the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. Okay, let's get started. The first question that we have here says, how well vascularized are the cardiac organoids?
Yeah, that's a often asked question actually. So the microtissue is about 200 to 300 microns in size, with about 5,000 cells. So we adopted the protocol from Christine Mamry's group. As per our observation with the tunnel staining and cell death staining, we do see that the size of the necrotic core is kept minimal there. While the endothelial cells act much like a scaffold of the organoids. They don't form tubes, really form tubes to vascularize the entire organoids, but there is very much minimal cell death in these cardiac microtissues.
Perfect. The next question that we have here says, how is the viability between lipofectamine stem transfection reagent and the neon NXT electroporation working with iPS cells?
Yeah, so we worked with the application scientists at Thermo Fisher, and we have did quite a bit of testing as well. And we are very pleased to see that the neon NXT system has significant improvement in viability compared to the lipofec stem approach that we have used previously in the lab. But both will work if if you are really looking at clonal selection and you're just looking at doing clonal selection for your self-interest. But it will speed things a lot faster with the NXT system because of the improved efficiency and the knock-in efficiency as well.
All right, wonderful. Okay, the next question that we have here says, how might amino acids like asparagine activate the mTOR pathway and through what kinases?
Yeah, that's a great question. So asparagine does not directly act through a kinase, but it also involves other players that indirectly activate the mTOR complex 1. For example, asparagine could increase intracellular leucine or arginine levels through asparagine exchanges like ASCT2, LAP1 antiporter system, and then activate and enable the REC GTPase pathway. And leucine and some of these branch chain amino acids has been known to be potent activators of the mTOR pathway. So it activates, but in an indirect manner.
Okay, next question we have here says, direct and self-assembly approaches. What are some of the considerations and thoughts?
So with direct assembly approach, it's great because it allows you to control specific cell types and abundance. And it also allows the end user to have more customization and integration with the biomaterials. However, it limits in terms of scalability than the self-assembly approaches. And also most self-assembly approaches to cardiac organoids does not have the immune cell type. So from our point, we wanted to get and try to model the immune lineage as well. And these immune cell types often comes from a different hemogenic lineage, which we then decided to opt for a more direct assembly approach. The self-assembly approach resembles much the morphology and structure that closely resembles the natural development of the heart. It's often scaffold-free, and it allows cells to establish more of a native-like ECM signaling gradient, which is great. So in the lab, we are now currently adopting more of a hybrid approach.
Perfect. It looks like we have two final questions here. This next one says, are there evidence of cardiac teratomas with OSKM overexpression?
Yeah, so there is with sustained activation of OSKM, rather than partial reprogramming, the work by Thomas Braun's lab at the Max Planck Institute showed that the pluripotent teratoma-like structures are formed with the functional deterioration of the heart and loss of contractile apparatus with sustained activation of OSKM. But with short-term partial reprogramming, these adult cardiomyocyte can retain its reprogramming potential and de-differentiate into more of a cardiac fetal-like form. So you push it up the hill, but you don't push it for too long and not all the way.
Okay, perfect. We will go ahead and wrap up here with this final question. This one says, how does the cell undergo de-differentiation and differentiation while contracting?
Yeah, so in many cases, cells has to suppress contractility for plasticity to happen partially. They need to reduce excitation, contraction, coupling, change, and alter the calcium dynamics. And studies and others have shown that pharmacological inhibition of contraction through the use of beta blockers do enhance the proliferative capacity of cardiomyocytes. In the de-differentiated cardiomyocyte cell state, the cells tend to be more poised to undergo cell cycle reentry through a more disorganized sarcomere and a more blunted calcium handling.
Okay, great. Well, thank you again, Mick, for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everybody. Goodbye.